Hello again. It's quite a long time since we last did a reading from my book, What is Man, Adam, Alien or Ape? But we're going to recommence those readings today, and we're beginning with chapter 7. We've reached chapter 7 in our readings, and we come now to this chapter, which is entitled The Devil in the Details. You'll understand a little more what that signifies presently. But because this chapter is somewhat technical, I think it will be helpful to read the introduction to the chapter before we actually read the chapter text. So here's the introduction. In 1986, I debated head-to-head with Richard Dawkins at the Oxford Huxley Memorial Debate. During a coffee break, and after we had both made our speeches, he and a colleague, both biologists, explained to me that the genes of humans and chimpanzees were 98% identical. Proof, they claimed, that both had descended from a common ancestor. Fortunately, I had enough biological knowledge to reply that however similar the genes might be, they are clearly expressed very differently in the two species. They had to agree. And herein lies a profound mystery. We now know that a single gene can code for hundreds of different proteins, so that the characteristics of any species depend primarily not on its genes, but on the marvelous control mechanism that dictates how those genes are expressed. We take a close look at one of the major mechanisms called alternative splicing, in which complex molecular machines decide what sections of the gene are required to make a given protein. They remove the unwanted portions and link the needed pieces back together. So let's begin the chapter. The Devil in the Details. Subheading, Digging Deeper into Genes and Genomes. We have two chapter heading quotations. The first is from Craig Clevenger, who invented the expression, the devil is in the details. He writes, by selecting a subset of descriptive details from a larger body of options, we can frame our descriptions in such a way that we cue the reader's imagination in a desired direction. And the second chapter heading quotation is by Franklin Harold, Professor Emeritus of Biochemistry at the Colorado State University. And he writes, "We must concede that there are presently no detailed Darwinian accounts of the evolution of any biochemical or cellular system, only a variety of wishful speculations." So the chapter begins. The first of these two quotes comes from an article on creative writing, but it has far wider significance than the author perhaps intended. It can readily be applied, for example, to political pronouncements, where the use of selective information is better known as spin. Selective information also turns up in scientific claims and publications, not least in the contention that humans and chimpanzees both owe their existence to a common ancestor, and ultimately to some primeval microbe. So we are going to take a close look at the use of selective data and ask whether or not the biological facts actually support the evolutionary paradigm. Biochemist Franklin Harold, who provides our second chapter head quote, clearly has his doubts. But before we can proceed, we need to understand a little more about the molecular basis of life. This won't take long, and you can skip this section if you already know it all. Subheading: Molecular Genetics for Beginners. The information needed to build a living organism, the genetic blueprint for its construction, is stored on the DNA molecules found in its cells. DNA molecules are long strings of chemical groups called nucleotides, and each nucleotide contains a smaller group of atoms called a base. That smaller group sticks out sideways from the string. If it helps, think of a daisy chain, in which each daisy is a nucleotide, and the end of its stalk is a base. Under most conditions, two such strings or strands of DNA combine to form a double helix. This This can be pictured as a spiral staircase in which the two nucleotide strands form the support rails and the bases protrude inwards to form the steps. Each step, therefore, consists of two bases, one protruding from each support rail, and the two bases join together in the center of the spiral to form a complete step, which is called a base pair. The two base half steps are linked together by so-called hydrogen bonds, which can be easily broken and remade without affecting the integrity of the DNA strands themselves. This allows the two strands of the double helix to be pulled apart temporarily or permanently in cell division to expose unpaired bases, which then collectively provide a template that can be copied or transcribed onto another kind of long-chain nucleic acid called RNA. The R stands for ribonucleic acid. These RNA molecules then carry the information copied from the DNA to various regions of the cell to facilitate or promote processes such as protein manufacture. That's why this particular kind of RNA is called messenger RNA. So how is the information stored? It's stored in the arrangement of bases along the DNA molecule, which contains 4 different bases, abbreviated C, G, A, and T. And these can be lined up along the DNA molecule in any order. However, their different sizes mean that C can only fit together with G to form a base pair, while A can only fit together with T. This means that there are only 4 different kinds of step existing in the double helix, namely C to G, G to C, A to T, and T to A. These 4 steps constitute a kind of 4-letter alphabet, which allows information and instructions to be stored in exactly the same way that a human language uses sequences of letters to form words, which in turn are organized into sentences that store and convey information and instructions. The DNA molecules are packaged along with special proteins into the chromosomes, which become visible under a microscope just before a living cell divides into 2 cells. As mentioned in the previous chapter, the totality of an organism's DNA is called its genome, while genes are those stretches of a DNA molecule which contain the information needed to make proteins. We say they code for proteins. Subheading: Misleading comparisons. Misleading comparisons. Let's now return to our main concern, namely the similarities between the chimpanzee and human genomes. Similarities that undergird the evolutionary claim that the two species have descended from a common ancestor, which lived between 6 and 13 million years ago. In the previous chapter, we saw that the chimpanzees and humans share 96% of their genes, and perhaps as little as 70% of their entire genomes, although a higher figure of around 81% is more widely accepted. We also noticed that gene sharing of genes between species, is virtually universal, and that humans share 88% of their genes with mice and even 18% with Baker's yeast. In evolutionary theory, this is held to be strong evidence for common descent, but all it really proves is that certain genes and gene types are essential for life of any kind, with greater similarity existing between organisms with similar needs. Birds need to fly, and fishes need to swim. However, in addition to this general conclusion, we must now look more closely at the similarities reported between genes in different organisms, especially chimpanzees and humans. These reports can be misleading for several reasons, and we'll explore some of them in this chapter. The first reason that the information we're given is misleading is selective bias. As an example, if you wanted to compare what kind of transport people prefer in London and New York, respectively, you would not limit your survey to pedal cyclists. Yet the methods used to compare genes and genomes involve highly selective procedures, in which only those DNA segments that are broadly similar in the two organisms are taken into account. The sections of DNA that bear no resemblance to each other are simply ignored and dismissed as junk, J-U-N-K, junk DNA. The supposed detritus of aeons of evolutionary trial and error. However, the idea of junk DNA has itself been junked following closer study of the human genome, particularly in a worldwide cooperative project known as ENCODE, E-N-C-O-D-E, the Encyclopedia of DNA Elements. This project was launched in 2003 with the purpose of identifying all the functional elements in the human genome, and it provides strong evidence that much of the so-called junk DNA does, in fact, have a biological function. I'll have more to say about that later. The geneticist Jeffrey Tompkins explains the problem of selective methods of gene comparison as follows. Quote, a scientist just published a study describing chimp DNA mutation rates and compared a number of cherry-picked genomic regions to human. And this research doubled their evolutionary timeline. So the researchers then compared selected DNA segments between chimpanzee and human that were highly similar, omitting the many non-similar regions. The scientists only used the regions that were about 98% similar and essentially threw out everything else. These are the regions that the researchers stated, quote, can be aligned with high confidence, close quote. It appears that all the dissimilar DNA regions get tossed out because they didn't fit the evolutionary paradigm, close quotes. To be fair, the work cited was only concerned with the speed at which genetic changes would have occurred, assuming that chimp and humans have both descended from a common ancestor. If that assumption were correct, it should not matter which DNA regions are compared, so that cherry picking for convenience becomes permissible. However, and this is the important point, it is a very different matter if selective comparisons are used to support the assumption that common descent has occurred. Elsewhere, Tompkins and Bergman provide a detailed technical review of the common claim that the human and chimpanzee genomes are nearly identical. They found these claims to be highly questionable based on an analysis of the methodology and data outlined in an assortment of key research publications. Well, how is it done? That's our next subheading. And geneticist Richard Buggs explains the procedure simply. To compare the two genomes, the first thing we must do is to line up the parts of each genome that are similar. When we do this alignment, we discover that only 2,400 million of the human genome's 3,164 million letters align with the chimpanzee genome. That is 76% of the human genome. Some scientists have argued that the 24% of the human genome that does not line up with the chimpanzee genome is useless junk DNA. However, it now seems that this so-called junk DNA could contain over 600 protein coding genes and also code for functional RNA molecules. Dr. Bugs goes on to explain that this is just the start. Artificial gaps have to be inserted into one or other of the genomes in order to properly align the matching regions, which introduce a further 3% difference. Other effects of a smaller kind are also found. Added together, these considerations reduce the overall similarity between the chimpanzee and human genomes to around 70%. 70%. The common assertion that the human and chimp genomes are over 98% similar clearly must be treated with skepticism. The truth is, of course, that regions selected because they are similar are, in fact, 96% similar. True, but tautological. To enlist this observation in support of common descent comes close to circular reasoning. Of course, evolutionists can claim that these similar regions, being rich in genes, are the only ones that matter. But this requires them to dismiss as redundant the 24% of each genome that is unique to its species, leaving us wondering why humans are so bad at climbing trees. To say that this 24% can be safely ignored because it is only junk DNA denies the results of ENCODE, which tell us that non-coding DNA plays a vital role in controlling the way that genes are expressed. More of that later. I then have a subheading, which is B books, and you'll realize what that's all about in just a moment. As noted at the beginning of this chapter, selective comparisons have persuasive power. They can cue, C-U-E, they can lead the reader's imagination in a desired direction. Selective comparison of genomes greatly increases the apparent similarities between two organisms, and thus gives traction to the theory of common descent. So let's try to illustrate the dangers of drawing conclusions from this kind of comparison. Suppose you are comparing two modern books by different authors on, say, beekeeping. Let's call the books Perfect Pollinators, PP, and Busy Bees, BB, respectively. You notice that there are certain similarities between the two books, and conceive the idea that both books were plagiarized from an earlier untraceable tome. You want to prove your point by carefully comparing the words and sentences in the two modern books. Starting with words, you find that 70% are common to the two books, whereas they share only 50% of their words with Margaret Mitchell's 1936 novel Gone with the Wind. You attribute this similarity to the existence of a common source for PP and BB, ignoring the fact that the two authors are dealing with a common subject and are thus likely to use similar vocabularies. You then get to work on sentences, but this proves more difficult. For example, BB has a chapter on beehive construction, whereas PP doesn't cover this subject at all. So there are no comparable sentences dealing with this matter. So you decide to compare only the 5% of those sentences in the two books that, while differing in detail, nevertheless do closely resemble each other. You conclude that both modern authors have lifted these sentences from an unknown common source, and you submit an article to the Journal of Beekeeping accusing both authors of plagiarism. Not surprisingly, the journal rejects your conclusions, pointing out that all your data can be better explained by the similarity of the subject and the identical desire of two independent and original authors to document their passion for bees. The illustration applies to genomes because triplet sequences of letters in the DNA 4-character alphabet can be regarded as 3-letter words, each of which codes for one of the 20 different amino acids used to assemble proteins. Then, just as in human language, the sequence of these DNA words, the order in which they appear, spells out a sentence which specifies which of the thousands of proteins found in living cells will be made by copying that particular gene. While no illustration is perfect, our beekeeping parable should warn us that the observed similarities between chimp and human genomes, the observed similarities between chimp and human genomes are far from proving the existence of a common ancestor. Next subheading: mixed up or incongruent genealogies. A second problem emerges when the genome comparison is widened to include other great apes besides chimpanzees. Evolutionary geneticist Ingo Ebersberger and co-workers sum the matter up in this way. Quotes, in 2/3 of the cases of gene comparison among great apes, a genealogy results in which humans and chimpanzees are not each other's closest genetic relatives. The corresponding genealogies are incongruent with the species tree. In accordance with the experimental evidence, this implies that there is no such thing as a unique evolutionary history of the human genome. Rather, the human genome resembles a patchwork of individual regions following their own genealogy. Note well the phrases. Note well the phrase, quotes, no such thing as a unique evolutionary history of the human genome, close quotes. As far as I can see, the conclusion would be equally well stated by leaving out the word unique. For since different parts of the human genome appear to have different evolutionary histories, there seem to be only two options. Either man was somehow created by stitching together genetic material from a variety of ape-like creatures, known and unknown, or else his evolutionary history is a fragment of evolutionary imagination. Our beekeeping analogy can be extended to illustrate this point. Suppose you are comparing not just two modern books, but say 5. Call them PP, BB, XX, YY, and ZZ. You continue to believe that they are all plagiarized from an early unknown work because of similarities between the word counts and sentences. However, as you examine the details, you realize that one chapter of PP might actually have been plagiarized from XX and another from ZZ. They're all modern books. In fact, PP seems to be a patchwork of plagiarized passages from the other 4 books, and even from one or more unknown source. Contrary to your original belief, PP might therefore have little or no connection with the first hypothesized unknown source. Your head begins to ache, and you decide to abandon beekeeping and take up stamp collecting instead. As Franklin Harold concedes in our chapter heading quote, there are no detailed Darwinian accounts of the evolution of any biochemical or cellular system, only a variety of wishful speculations. Well, we're going to leave it there for this session. We'll continue with this chapter in the next one. Thank you.